Okay, so we continue onwards now with our aerobic pathway. So we get into now our discussion of cellular respiration. So up to this point, we have covered two of the three catabolic pathways. So we know that we have this sort of light blue arrows that represent glycolysis, where we convert glucose into pyruvate in the cytoplasm. If pyruvate finds itself in an anaerobic condition, no oxygen present, glucose then progresses into the fermentative pathway. So pathway number 2, sort of whitish arrow, grayish, light gray arrow. So pathway 1, glycolysis. Pathway 2, if anaerobic, we go into the production of lactic acid for humans. End of the story. Right? Now, if we backtrack, we have our pyruvate, we go through pathway 1, and we have oxygen available, then we'll progress into the mitochondria for cellular respiration. So again, there are a total of 3 pathways. The cell will always progress through only 2, either glycolysis and fermentation, if no oxygen, or glycolysis and cellular respiration if oxygen is present. So depends on the availability of oxygen, whether or not we progress to the anaerobic pathway or to the aerobic pathway. If there is no oxygen available, we go into fermentation. If there is oxygen available, we go into that mitochondria and cellular respiration, the aerobic pathways there. So this is a very zoomed out, very simplified view of cellular respiration. And I'm going to come back to this at the end of our whole discussion, but this is summarizing from glycolysis to the end of cellular respiration. So we have glucose with aerobic conditions, and all the enzymes, all the steps, everything is going to get converted into 6 carbon dioxides, 6 waters, 36 ATP, and an undetermined amount of heat. And so I'm going to come back to this, and we'll look at all these numbers and details at the end. But when we talk about cellular respiration is defined into 2 or 3 little sub-pathways, right? Some people maybe combine these 2 together. I'm going to break it up into the 3 little sub-pathways. We're going to call these pyruvate oxidation, the citric acid or Krebs cycle. Krebs was the guy that discovered it, so they give him credit sometimes. And the electron transport chain. So pyruvate oxidation plus citric acid cycle plus the electron transport chain all together equal cellular respiration, which happens only in the presence of oxygen. So, and again, sequentially, this has to happen after glycolysis, and it happens not in the cytoplasm, but inside of that mitochondria. So important. So if you can just kind of analyze the name, it gives you an idea of what's happening here, right? So pyruvate oxidation, we're going to oxidize pyruvate. We're going to strip electrons off of pyruvate. Citric acid, maybe not too informative, but cycle. That means we're going to do a lot of steps and we're going to end up back where we started, right? So we start here, we do a lot of things, but we get back where we started there. And then the electron transport chain, we're going to transport electrons. And you should know of a particular molecule that's actually transporting the electrons, right? So again, they are sequential, right? They do follow along, but they're always constantly going. So there is some overlap, and it's just a lot of things happening at the same time here, right? But it's a busy process that we term as cellular respiration. So let's look at pyruvate oxidation. Pyruvate oxidation, a pyruvate is oxidized in the mitochondrial matrix, deep inside the mitochondria, right? We lose a carbon dioxide, acetyl-CoA molecule is formed. What is acetyl-CoA? It's an acetate attached to a coenzyme A. and we generate an electron transporter. And since we have two pyruvates, remember we broke one glucose in half, so we have two pyruvates. This process is repeated twice for the second pyruvate that comes in. And if you look at that, I hope that makes sense. And I remember when I was learning this stuff, this didn't make sense to me. Like, I don't know, I don't know too many names. I'm lost where we are, the physical location, the sequence of the whole mess. But if I can try to explain it in a different way, hopefully this helps out a little bit. We just kind of progressed through Halloween, right? The Halloween season. And I don't know if you're fans of scary movie stuff. Right? Some of you are fans of Jason or Michael Myers or Chucky, or one of my favorite was Freddy, right? Pyruvate oxidation reminds me of scary movie stuff. Right? In scary movies, things are getting chopped off. Heads are getting chopped off, and things are getting split apart, broken apart. And it's the same kind of concept here, right? So instead of heads getting chopped off, let's look at it as a carbon dioxide getting chopped off. So scary movie stuff, right? So we get into the idea of, I'll come back to that in a bit, of scary movie stuff. So here we start off with our 3 carbons, 1 pyruvate, and in all scary movies, it starts to say, right? Oh, they should not be at the lake at night. They should be at home, right? But they go to the lake. They go into that old little shack or something, and they open the door. So 1, 2, 3 carbons enter, and they go in through the outer mitochondrial membrane, through the cristae, to the inner mitochondrial membrane, and then into the matrix. And then bad things happen in the matrix. So the first thing that happens, we get the head chopped off. We get carbon dioxide chopped off. So this bond right here is broken. So we lose a carbon and 2 oxygens. Carbon dioxide is broken, split off. So we basically have like a headless chicken, right? These 2 carbon molecules are floating around. Electrons got released. So NAD plus is there to pick up an electron to form NADH plus H. This headless chicken, these 2 carbon molecules are then going to get picked up by this molecule we call coenzyme A, right? Coenzyme A picks up the 2 molecules, and we now have this molecule we call acetyl-CoA, right? So think of coenzyme A. I like to think of coenzyme A as Aztec guy, right? I don't know if you know the mythology behind the scene, but Aztec guy, this strong guy, picks up this lady, carries her on, right? Carries her over. So she's not just going to lay there. She's going to get picked up and moved to another location. So Aztec guy, right? What's his name? Coenzyme A, right? And when Aztec guy combines with the 2 carbon molecules, we call him, that's a fancy Aztec name, no? Acetyl-CoA. Right? Acetyl-CoA is this Aztec guy with the maiden, and we have now this transporter of carbons. We've talked about transporter of electrons, which is NADH. Now we have a transporter of carbons, which is this coenzyme A, acetyl-CoA kind of molecule there. So all this happens with 1 pyruvate, but then the second pyruvate comes in and the same process happens. Whoosh, scary movie stuff, head goes chopping off, carbon dioxide's gone, NAD plus picks up electron to form NADH, coenzyme A connects with the 2 carbons to form acetyl-CoA. So per 2 pyruvates, we then have, per glucose, 1 glucose, 2 pyruvates, we then have 2 carbon dioxides, 2 NADHs, and 2 acetyl-CoA molecules, right? And that's what we call then again pyruvate oxidation. So pyruvate oxidation, we had pyruvate that formed in the cytoplasm. Pyruvate enters through cristae, I should say through outer mitochondrial membrane, cristae, inner mitochondrial membrane, and matrix. It's going to show it in the wrong spot. It should actually be inside the matrix where all that is happening. So again, should be actually in here. But let me pause that there, and I'll shift to the next sub pathway, which is the Krebs cycle.